The overall goal of this procedure is to embed yeast colonies. This is accomplished by first removing a colony and the underlying agar from an agar plate, placing it on several drops of molten agar, and quickly covering it with more molten agar. Next, excess agar is trimmed from the colony to make it small enough to fit into a mold. The agar-embedded colony is then fixed, stained, dehydrated, and finally infiltrated with spurs resin. The block is then placed in a mold with additional spurs resin, incubated until hardened, and then sectioned. Ultimately, results can be obtained that show the distribution of cell types within a yeast colony through light or electron microscopy.
The main advantage of this technique over existing methods like scanning electron microscopy is that it allows the internal structure of the colony to be determined.
This method can help answer key questions in the field of microbial development, such as the distribution of cell types within colonies.
This method provided insight into the structure of Saccharomyces cerevisiae colonies, but it is likely that it can also be applied to other microorganisms.
Visual demonstration of this method is critical, since handling and embedding of colonies may be difficult to learn from a written protocol.
The idea for this method was inspired by some genetic data we published in 2002 that suggested that spores were not evenly distributed in colonies. We adapted a method for sectioning colonies that was developed by Engelberg in the Fink lab.
So let's get started.
Begin by incubating wild S. cerevisiae yeast on agar plates at 30 degrees until approximately 300.